Good morning, everyone. Thank you for coming out to listen to me. I hope I make your time worthwhile here. I'm Mike Dombeck. I lead business development for Precision. Precision is a gene editing company dedicated to improving life through healthier food, overcoming cancer, and curing genetic diseases. At the heart of Precision is our Arcus genome editing platform. Arcus is a homing into nuclease-derived non-CRISPR editing system developed entirely at Precision, something that we have complete freedom to operate with. We apply Arcus across these three business areas: cancer immunotherapy, our healthy donor-derived allogeneic CAR-T platform, our gene therapy, in vivo gene editing therapeutic applications, and our food and agriculture group. 2018 has been a significant year for Precision. Earlier this year in June, we announced a $110 million Series B financing to advance our therapeutic programs. We followed that in July with the first industry publication demonstrating efficient, durable, non-toxic therapeutic editing in non-human primates. That was work Precision did with Dr. Jim Wilson at the University of Pennsylvania. And most recently, a few weeks ago, we followed that with our announcement of partnership with Gilead to pursue in vivo gene editing as a potential therapeutic to treat hepatitis B. This is a program that Precision will lead all of preclinical development. Gilead will take over clinical development and global commercialization, as well as full funding for the program. We'll be following these milestones up in the not too distant future, we hope, with our first IND for our lead CAR-T program, as well as early next year bringing online our own in-house GMP manufacturing for mRNA, AAV, and our CAR-T programs. So turning to Arcus and the core of our technology, we believe that Arcus represents the therapeutic-grade genome editing platform because of a number of features that distinguish it from the other gene editing technologies you may be familiar with. The first that I'll reference is our safety feature. Arcus has a natural off switch. In its resting state, it's not capable of cutting DNA. Arcus must bind productively to its target site on DNA, upon which it undergoes a conformational change that actually creates the active site of the molecule, allowing it to cut the DNA. If there's no active site, Arcus is not capable of cutting DNA, which significantly reduces any risk of random off-targeting damage to DNA. Second is delivery. Arcus is a single protein encoded by a single gene. It is under 1,000 base pairs. What this means is that we can fit multiple Arcus nucleases into any of the common delivery systems, viral or non-viral. Turning to efficacy, number 3, Arcus works in a very different way from the more familiar mechanisms of the other gene editing technologies. Arcus does not rely on base pair matching and a guide sequence. Arcus instead works through electrostatic interactions of the amino acid residues of the Arcus molecule to the DNA backbone. What this means is that Precision has all of those amino acids to work with to precisely control the affinity and cleavage activity of each Arcus nuclease. Essentially, we can customize each nuclease to the specific application that we intend to use it for. Fourth is repair. Arcus is unique in the type of cut that it creates in DNA. It is the only gene editing technology to leave a 3' overhang when it affects a cut in DNA. This is important because in eukaryotic cells, a 3' overhang is required to initiate homologous recombination. By leaving this 3' overhang, we can drive efficient repair with a donor template through the homologous repair mechanisms present in a cell. And then finally, differentiated on targeting. Precision is going on 13 years old now. We have worked in plants, animals, and human cells. We have yet to find a target in any of those that we have not been able to hit. What underlies that differentiation for Arcus is that we are, as I mentioned, we're not dealing with a guide RNA or base pair matching. What we do at Precision is protein engineering. as I mentioned, we work with modifying those amino acids. This takes a long time and a lot of expertise. As you see at the bottom of the slide, to generate an initial Arcus first-generation nuclease to a target takes us up to 5 weeks to do the protein engineering to develop that. We will then put every nuclease through multiple generations of optimization to control that binding affinity and cleavage activity, which takes up to 6 months. But at the end of that, what you come away with is a therapeutic grade clinical candidate. The way we do that is through that protein engineering and control of those amino acids. You can see here the colored areas are what we go through designing to control the precise affinity and binding activity of the nuclease to make it specific to its target and optimize that activity. The complexity of this also underlies our IP portfolio. The Arcus platform, as I mentioned, we have full freedom to operate. We have over 45 issued patents protecting the platform alone. In addition, the design of Arcus nucleases are not fully patented. Elements of it are treated as trade secret. By treating these as trade secret, each Arcus nuclease is a non-obvious entity. That means each Arcus nuclease is also patentable in and of itself as a new drug substance or product. So again, we're applying the Arcus nuclease technology across 3 business segments: our cancer immunotherapy, genetic and infectious disease, and our food and agriculture programs. Turning to the cancer immunotherapy, our application here is, as I mentioned, in healthy donor-derived, off-the-shelf allogeneic CAR-T. Our strategy here is to advance our lead program, a CD19 program, into the clinic to de-risk the platform and then rapidly expand into other CAR-T targets. The core of our approach to healthy donor-derived CAR-T is a single-step editing and engineering process to create our CAR-T cells. We deliver our Arcus nuclease targeting a knockout of the T-cell receptor in combination with a CAR-T sequence that has been designed to match the break we create in the T-cell locus. The result is that in one step, we knock out the T-cell locus, creating the allogeneic cell, and target the insertion of the CAR in its place. The result is a consistent, predictable, potent drug substance. We have taken this process and scaled it up to full-scale manufacturing from a single donor, from a single donation from a single donor. We are currently producing about 200 doses. The lead program here, as I mentioned, is a CD19 program. We anticipate filing the IND for this program before the end of the year. And then one of the programs that we'll be following that is our anti-BCMA program. Our anti-BCMA is taking a very different approach than the other CAR-T BCMA programs that you may be more familiar with. Our strategy here is to leverage our manufacturing efficiency and the associated low cost of goods to produce this candidate to target populations that currently wouldn't be appropriate for CAR-T and are addressed more by traditional therapeutics such as antibodies and chemotherapy, such as pre-transplant conditioning, post-transplant consolidation, smoldering myeloma, and other populations. Turning to our genetic and infectious disease, similar to the CAR-T program, our goal is to take our most advanced programs and rely on them to de-risk the platform more broadly before pushing into a more extensive scope of targets. Leading the way for this is our hepatitis B and PCSK9 research activities. I mentioned earlier that previously this year in July, we had the first publication of in vivo gene editing in non-human primates. That was a PCSK9 knockout, again done with Jim Wilson at University of Pennsylvania. With that, our first cohort is now out past 2 years since their first treatment. This was a single dose administration. We continue to see that this is durable, effective, and well tolerated. Turning to the HPV program, we've demonstrated with this that Arcus nucleases can eradicate HPV viral DNA from primary human hepatocytes in vitro. This program is now partnered with Gilead in a worldwide development and commercialization license. Precision is responsible for drug formulation and research throughout preclinical, followed by Gilead taking over the IND, clinical development, and full worldwide commercialization, and Gilead will fully fund the program. Following on those, we have two other programs I'd like to mention today. The first is our Retinitis Pigmentosa program. This is a program that has achieved proof of concept in a mouse model demonstrating photoreceptor morphology rescue. In that model, this is something that has moved into large animal studies in a relevant pig model. And then I'd like to also talk about our Hemophilia A program. This program, we believe, is unique in the industry in that it does not require a donor template to produce factor VIII. What we are doing here is targeting the Hemophilia A population that has the intron 22 inversion. What we are doing is delivering an Arcus nuclease that creates a break and a repeat associated with that inversion and attempting to recreate the event so that we take the inversion and restore it to the proper wild type orientation. We have done this in vitro in patient cells and demonstrated that we can convert with one administration 30% of the cells to the proper wild type orientation and wild type natural factor VIII production. This program is also in the relevant dog model right now with a canine model at Chapel Hill. Finally, turning to our third business segment, food. We are committed here to pursuing human wellness and food security through partnerships with established multinational food organizations. Our lead program here is partnered with Cargill. It is a low saturated fat canola oil that recently moved into field testing with Cargill. And the final product here will be considered a non-GMO product. In conclusion, 2018 was a significant year for Precision. Before the end of this year, we also do anticipate filing our IND, as well as having large animal proof of concept data across primates, pigs, and dogs, all three of the relevant large animal models, coming up in 2019, moving into our first clinical trial and bringing online our own in-house GMP manufacturing. So we're looking forward to continuing our success in 2019 and the future. With that, I have a little over 2 minutes left. I'm happy to take a question or two. Yes? Absolutely. So for our CAR-T programs, the process doesn't change. All that changes is the target that we are engineering into, the CAR sequence that we're engineering into that TCR locus. So with the process not changing, the efficiency remains the same or improves as we refine the process.
How are you rendering these cells allogeneic?
We knock out the T-cell receptor with the Arcus nuclease. All right. Thank you.